Everyone. My name is Monica. I am the head of go-to-market at MiLaboratories. So today's webinar is a joint collaboration between MiLaboratories and Miltenyi Biotec. We have today our presenters, Jana Henk, who is the global product manager of Miltenyi, and we also are joined by Sam Pozlowski, who is the CEO of MiLaboratories. So today's topic will be all about streamlining TCR alpha beta discovery. So we'll be taking you through the entire workflow from the wet lab all the way to the analysis and insights. So a really exciting topic today. Just some housekeeping items. If you have any questions, feel free to drop it in the chat or save them to the end because we'll have a few minutes for questions. And also, the recording will be sent tomorrow morning. So if you're unable to make it, just be on the lookout for an email from me so you can watch the recording. So jumping in today's agenda, Jana here will first talk a little bit about NGS, talking a little bit about the sequencing, the kits, and then we also have a live study. And then Sam will also move into the insights from the study using a live demo of Platforma. So now I'll hand it over to Jana.
Thank you, Monica, for the kind introduction, and also from my side, a warm welcome to everyone joining today. I'm a product manager from molecular analysis products at Miltenyi, and I want to briefly introduce Miltenyi with some key facts before jumping into the topic. So Miltenyi is a company founded 37 years ago, with grown globally to nearly 5,000 employees worldwide today. And what I find remarkable is nearly 1/4 of all employees are working in global R&D, which allows us to constantly develop and innovate products to help cutting-edge research and treatments against diseases. As Monica already mentioned, I will start with a general overview of library preparation since we have a audience with a broad range of knowledge about that, and then move into bulk versus single-cell TCR sequencing, then move into the kit, which is a TCR alpha beta RNA profiling solution, then talk a little bit about the case study example, and then hand over to Stan, as Monica already said, who will talk about the analysis solution Platforma, bulk TCR data analysis with Platforma, and then we have time for some Q&A. So jumping right in, the general RNA library preparation workflow, I think about it in three distinct steps. One, of course, is the sample input. We want to have high quality RNA isolated from a tissue of choice or of interest. And then this RNA is turned into adapter ligated or added cDNA from this RNA. And it has various different subsets, right? The RNA gets fragmented, reverse transcription into cDNA. There's end repair. We have definitely different cleanup steps in there, and adapters are added to have them fitted to the sequencer of choice. Then the library is amplified to have sufficient yield for the sequencing, and after successful quality control, which is also very important for the workflow, it can go into sequencing. For sequencing, which we will talk about today, is Illumina-based. It highly depends on your interest, how the length is determined. For example, when we look at the T-cell receptor, if you only look at the CDR3 region there, you have 150 base pair paired end sequencing is sufficient. But if you want to look at the whole receptor, you want to apply 300 base pair paired end sequencing. The general goal of RNA library preparation and sequencing and the data analysis is to look and compare the transcription profile and the average expression levels of genes in a large population of cells or tissues. And then, if you have multiple samples, to perform differential expression analysis to study, for example, a disease mechanism, to identify biomarkers, or simply look at the differentiation of a sample or developmental questions. The application is really wide. What is a little bit more narrow is if we look at immune repertoire library preparation, right? I just explained the general workflow, and there are two major differences when looking at the immune repertoire library preparation. One is the sample input. We are talking about immune cells here, so it should be PBMCs. In our case, it can be T cells or RNA from immune cell containing tissues. The second very important step is to use specific primers, because if you don't use specific primers to get the target of interest, you have a high risk of getting off-target molecules within your library and then waste reads in sequencing, which will also make data analysis and the cleanup in data analysis a little bit more work. So specific primers are really of top priority here. For data analysis, I put Platforma here because our solution has a preset with Platforma to enable researchers to go from input, library generation, sequencing to their own data analysis, even with little experience in coding. How do you apply immune repertoire sequencing? Well, if you have questions about your immune repertoire diversity, or if you want to identify clonotypes that are associated with a disease or a treatment or biomarkers, this is the right way to go. And also, if you span a longer timeline, you can track the immune response over time. Now I want to go into a little bit of a comparison here between bulk TCR and single-cell TCR sequencing. We at Miltenyi are fans of bulk TCR sequencing, and also for a good reason, but I want to emphasize here that these are distinct methods, right? None of them makes the other one redundant, and both have their separate applications. For bulk TCR sequencing, these are, for example, in-depth repertoire information and a cost-effective experiment. You get a robust quantification of clonotypes out of it because it's the average expression across a sample. Whereas for single-cell TCR sequencing, you get the resolution of the T-cell receptor expression per cell. So you have immune cell subset analysis because you have the resolution of the single cell. You can do direct alpha-beta chain pairing, and you can, in most methods, connect the T-cell receptor to the cell transcriptome. Conversely, there are also disadvantages. We have limited alpha-beta pairing information when looking at bulk TCR sequencing, although some computational methods might bridge that knowledge. And there's no link between the T-cell receptor to the transcription profile of the sample. For single cell, we have a high cost per sample and a high sample quality requirement as input, and there's limited repertoire depth. For applications, as I said already, they differ quite a bit. Two examples here might be that you want to apply bulk TCR sequencing to get a cost-efficient, high sensitivity overview of the immune repertoire changes in a tissue or a population that you're interested in, whereas you want to apply single-cell TCR sequencing to identify distinct immune cell subsets because you have that resolution, and you can link the receptor expression to the transcriptome of cells. I already mentioned, Miltenyi Biotec has just recently launched a bulk RNA TCR alpha beta profiling kit, and we did so with great resolution by combining high sensitive multiplexing primers with unique molecular identifiers. I want to talk a little bit about key features here. One is that we have a preset with Platforma enabling researchers to go from library generation in data analysis because the preset is tailored to our chemistry. Second, we have a straight from cell option to allow flexible input. So if you have PBMCs or T cells, you can skip the prior RNA isolation and directly jump into library generation. Also, because we emphasized optimal primer design, our kit can work with a wide range of input RNA, starting as low as 0.1 nanogram until 500 nanogram. Although only shown to 200 nanogram here, the solution provides robust clonotype output across the complete range. Our very talented R&D team also made sure that the experimental setup is robust and tested for operator-independent reproducible results. So here you can see the clonotype counts of two distinct operator uses, who the operators use 25 nanograms of RNA on 2 different days in 2 different settings and generated 2 libraries with 2 replicates each. And they also have different knowledge of library preparation in the lab work, and they retrieved distinctly similar clonotype counts for TCR alpha and for TCR beta respectively. Last but not least, it's super important to us that you can use this kit for generating cutting-edge science research, and that sometimes means you have to work with complex samples. And we are happy to show that our kit enables high clonotype capture exceeding the results of other commercially available products, here shown for 250 nanogram of FFPE RNA as input. So to summarize, our newly launched TCR-Profiling solution enables highly sensitive and robust T-cell receptor clonotype analysis with flexible workflows. But not only I'm of that opinion, but also these features were recently used by a study which is currently on bioRxiv. They took advantage of the fact that our kit is reproducible and highly sensitive, and also, of course, because bulk TCR sequencing is cost effective. The researchers generated their data by applying the chemistry in combination with antigen-specific T-cell activation to develop an assay to discover antigen-specific clonal responses to COVID infection. As one example, I want to present one donor-tracked immune response who first received a vaccination and then over the course of 1.5 years got infected with COVID twice. The researchers captured the immune response, sampling PBMCs over 10 different time points, shown here, and performed T-cell receptor bulk sequencing in 2 technical replicates, analyzing the clonotype expression during this time. I'm only presenting a tiny fraction of the results, and I recommend you to check out the publication in total because it's really fascinating results. But by stimulating with COVID-specific proteins, M and M here and S here, they identified over 90 TCR beta clonotypes expanded during the infection events. Here only shown for the CD4 positive T cells. And what was most remarkable to me was the sensitivity of the assay, with most clonotypes presented at below 0.1% frequency in the samples. I want to briefly summarize here that they could span the complete timeline of vaccination and infection events. And because of reproducibility of our assay, they took 2 technical replicates were sufficient. And because of the high sensitivity of their assay, they identified low frequency COVID-specific clonotypes. And finally, they performed expansion-based TCR discovery analysis, and with analysis being the key word, I want to hand over to Stan to talk about the analysis Platforma options that you can do with bulk TCR sequencing. Should I stop sharing or could you just take over, Stan? Let me know.
I will share my screen. Yeah.
Perfect.
Okay. Well, thank you very much, Jana.
My pleasure. Thank you, Stan.
Okay, I've sent a request, Monica, to share my screen.
Yep, all good.
So you see my screen? No, not yet.
Great.
Okay,
Yep.
you see my screen now. Okay, again, thank you very much, Jana. So with this great introduction into the wet lab part, I'm switching to the data analysis because data generation is not the time when you stop with your research and you discover, and once generated those gigabytes of data, you need to do something with them to get the real biological insights from. Yeah, and today the analysis of next generation sequencing data, especially of the complex modalities like repertoire data, which is a pretty complex data, is tricky, both in terms of the data scale and the complexity of the algorithms and methods. So that's what we have in our platform, ability to get this done really fast and convenient. So questions which we typically ask when we're doing the TCR sequencing and studying the TCRs are pretty different. So we may look on the overall repertoire as in general, and asking what are clonality metrics of our repertoires, or what are the TCRs that are likely to bind to the antigen, what receptor sequences are specific to or differentially abundant in certain conditions, and track those across time points or different metadata values, etc. Those things we typically ask to the data when we're doing the TCR analysis, and specifically in the case that we're going to study today, that's one of the things that we're asking. What are those T cells that are involved in the immune response and activated with the COVID? So just a few words about our company. We are in this field of immune research for more than 10 years, starting with our first software tool, now product called MiXCR, which is a tool for TCR and BCR next generation sequencing data analysis. It's pretty widely used in academia and industry. So we have it's published and very well cited. So I think today it's the most widely used tool for NGS analysis of TCRs and BCRs in academia. And we have more than 1,000 of organizations actually running MiXCR on a daily and Platforma today on a daily basis and doing a lot of research. So today my focus is Platforma. It's the interactive platform where you can do the in-depth analysis using tools like MiXCR and advanced post-analysis tools within the zero-code environment with intuitive interface that actually speaks biological language. You don't need to write Python or R code. You can just get the results and the answers right from the data yourself. It's a very transparent tool. So when I'm saying that it is a zero-code environment for biology researchers, it doesn't mean that you don't need to do bioinformatics. Actually, Platforma is built on the very transparent bioinformatics algorithm validated by the industry and academia, and we have partners and researchers from academia and from the industry who are contributing to the platform, who bring in their bioinformatic tools for in-depth analysis of T cells and B cells. Yeah, so with this in mind, just a really brief overview of what are the relevant tools that we have on the platform to TCR discovery. So we have, of course, the clonotyping powered by MiXCR, and Miltenyi has a dedicated blog tailored to this chemistry, Miltenyi TCR alpha beta profiling kit. Yeah, we have tools for clonality analysis and various characterizations, clone tracking, powerful visualizations, and ability to integrate the different sources of the data, and advanced tools like, for example, specificity prediction, which is provided by some of our collaborators using top-notch AI and machine learning models. So with this, I'm going to switch to a live demonstration of the software. Just give me a second. Let me set up this. Okay. So let me share back. Okay, Monica, do you see my screen? Yeah, yeah, I'm sure. Yeah. Amazing. So what you see here is the live demonstration of Platforma. So first of all, this is a graphical user interface. That's the interactive GUI app that you can download and install on your personal computer and personal laptop. Specifically, what you see here is one of the key results of the study. This is the time tracking of the 27, specifically 27 T cell receptors that are specific to the COVID and were activated by the immune system in response to the COVID. Yeah, if you remember the time frame of the study, the T8 is the time point after the second COVID infection, and T3 is the time point after the first COVID infection. Yeah. So here, when we're looking on this time tracking analysis, we see that these specific T cells are activated at the peak of activation exactly after the second COVID event. At the same time, they were also activated slightly, but clearly activated right after the first COVID event. So how we get to this specific results and how we were able to get this specific result in Platforma. So what you see is the project where the analysis is already set. On the left, there is a sidebar with the blocks, or well, with the project structure, starting from the raw sequence and data FASTQ files towards this nice visual. And to build this project, we start from the samples and data. Let's just go from the very beginning, live demo, and show how we can set up the full analysis really quickly, starting from the raw FASTQ files and go directly to the biological results. So when we start a new project in the platform, the very first step is to import the data that we have. Yeah, and we may use different data, FASTQ files, FASTA files, or whatever. Well, specifically for this study, we're using the FASTQ files generated with the Illumina sequencing machine. Yeah, so we're selecting all of the FASTQ files available in the project. Yeah, we assign the name to this data set. Let's say TCR bulk sequencing. Yeah, TCR bulk. and we import. As you see, this tool, this import tool of the raw sequencing data automatically matches the sample name from the file name. So and pairs the read one and read two files from the raw data. So once we create the data set, we have samples and pair of ASCII file for each sample. Before running the analysis, actually, and to do and get the real biology from this data, we need to support our data set, our FASTQ files with the metadata. Yeah, and this can be done by just importing sample sheet or just metadata, Excel file or TSV file from your computer, for example. Yeah, so I have an Excel file where it's just written which sample corresponds to which time point. Yeah, well, in this specific case, let's go here. We have I have the metadata only for a specific donor. Yeah, so it's the donor that we're considering this study, and the key metadata that we have here is the time point. So this time point is the time when the blood sample was collected. What's important here that for each time point we have a couple of replicas, and this is one of the features of this specific study. This replicates actually allows us to perform the differential analysis and find specific clones of interest. So I click Run button to make this data imported, and then the next step of the analysis is the clonotyping. And for clonotyping with this TCR alpha beta chemistry, Miltenyi has a dedicated block available on the platform. It's called Miltenyi TCR clonotyping. And in general, you see that we have a lot of blocks on the platform dedicated to various types of biological analysis, yeah, for different omics, etc. Well, for the TCR analysis with Miltenyi kits, we're using Miltenyi block for TCR clonotyping. And as all of the blocks available on the platform, they are open sourced, so you can find the code for the block. You can find the pipeline and the documentation behind the block. Yeah, specifically, Miltenyi TCR clonotyping block is powered by MiXCR tool under the hood, which is very important. And the parameters of MiXCR are tailored specifically to the Miltenyi TCR chemistry. So when we add this block, right now we have the one option, TCR bulk and TCR alpha beta RNA profiling kit from human. We can click Run button and start the analysis. I'm not going to click Run button here because it will take a couple hours to process 100 gigabytes of data available in this project. It's a pretty big project. Yeah, so let me switch to the project where I already processed this data. And talking about the chemistry, it's extremely important what you will have here, because for every sample, this block will generate a comprehensive quality reports and garbage in, garbage out. Yeah, if something happened to the wet lab, you're not getting good results in the data analysis. And here, of course, it's a very nice study, so the quality is excellent. This is a very, very good quality of data, and that's not only the feature of the authors of this study, of the scientists, but that's also a feature of the chemistry of the Miltenyi TCR profiling kit. It's really very, very nice kit, and it's pretty easy to use and provide a really good quality. If you click on the individual sample, for example, this sample, we have some warnings. We can go to the quality checks and see the comprehensive biological quality checks, yeah, showing like what's potentially may be improved or what potentially may be wrong in the wet lab side. Like for example, here we have a like slightly higher percent of the barcodes collisions in the clone type assembly, meaning there are slightly higher than expected number of CDR3 sequences with, UMI sequences which correspond to different CDR3. So it's barcode collision. Yeah, if you go to deeper, and I know there are some bioinformatics scientists on this webinar, you can go to the, for example, MiXCR log. That's just the command line log that bioinformaticians see when they run the MiXCR analysis corresponding to the specific chemistry with all the comprehensive reports. Yeah, also what we have here, we have a QC report table overview with all of the statistics about each sample and what exactly went, what are the numbers there. So this is the first essential step for the analysis when we start with the FASTQ files, yeah, the pre-processing part or clonotyping part. The next question every scientist asks is how to look on the repertoire, how to just look on my clonotypes. I want to see them. Yeah, the number one question. So one way is to use Clonotype Browser. That's the app we have on the platform, which provides, I say, a boring view on the repertoire. It's a table. Here, there are two options. By sample, when you look on your repertoire, selecting sample. We have many samples here. We can select sample, and then we see the repertoire corresponding to this sample. We have the clone ID, the CDR3 sequences. In the columns, we have many other information. Of course, we have the abundance, and abundance in our case is measured in the number of unique molecular identifiers. Yeah, so we have some highly abundant clonotypes. We have some that are not, etc., etc. The other way is overlap. In the overlap, we have the whole set of all of the clonotypes from all samples. For example, if I sort by the number of samples, I will see that some of my TCRs are actually present in 20 samples, in almost all of the samples corresponding to this type. So this is one way to look on the entire repertoire. But there is a more powerful way to look the entire repertoire, more visual. It's done with the app, which is called Clonotype Space. So Clonotype Space, and I have it here, this project already processed. Clonotype Space is an extremely powerful instrument allowing to visualize the entire space of your TCRs on a UMAP plot. The idea of the UMAP plot is that two dots, each dot here corresponds to one clonotype, and two dots located closer to each other if they share similar properties. So in this specific study, we have millions of TCRs, and we are now looking on those millions of TCRs on this specific plot. We can configure this plot to layer different source of information here. And for example, what I did, I put the J gene as a color on this plot. But we can put other things, like we can put the V gene, yeah, or the total number of UMIs as a size, for example, yeah, on this interactive UMAP. What else we can do? We can use the lasso instrument to select a subgroup of our TCRs and then, for example, compare them on the multiple sequence line. And we will use this instrument to find the clones with the specific to our COVID, and we can use this instrument actually a lot to interactively explore our repertoire. So this is like just an example of how we can look on the entire space of our T cells. But let's return to this specific study and try to find the answers, what's going on with this patient and what are the T cells activated in their response to the COVID. How are we going to do this? Well, there are a couple of instruments. The first instrument that we typically use when we do the T cell analysis is the clonality. So we want to see whether there is a clonal expansion in the response to the event. The way how it can be done in the platform is by using repertoire diversity analysis block. It's already added in this project, yeah, and by default it calculates several diversity metrics, very standard diversity or clonality or richness or dominance metrics. Yeah, basically saying us how diverse the repertoire is. The defaults are good to go, and everything, just click run button, runs, and then we're getting the nice results. So let me go to the diversity graph. This section is showing us the diversity on the plot. By default, by the way, it will look something like this. Yeah, that's how this plot will look by default. But pretty ugly, yeah, in this specific case because we have long sample names. But the power of Platforma is that you can do a really extensive fine-tuning of the visualization. You can actually do the thing that you can do with Prism GraphPad. Obviously, many of you know Prism GraphPad. So let's change this visualization. Let's group our samples, not by sample, but by the time point. Yeah, by the time point. We're grouping by the time point, nice. And we can change the type of visualization from the bar plot to what I'm using here, line plus bin dose. Yeah. On the y-axis right now, we're looking on the Chow-1 estimate. Chow-1 estimate is the diversity metric. It's a diversity estimator, which gives us a theoretical diversity of the repertoire, number of species, number of unique TCRs, if we sequence to the full depth. And what we see here immediately that at T6, we have a really diverse repertoire. Yeah, so the diversity is high here. Yeah, when we're looking on the Chow-1. What does it mean? It means actually that at this time point, the depth of sequencing was much higher compared to the other time points, and we can verify this on, for example, rarefaction curves. And on the rarefaction, you see T6. T6, that's our time point. Let me change the color of this plot to make it like, for the T6 time points, we see that the rarefaction curve actually goes higher, meaning that the diversity in terms of sequencing, if we sequence more, we have high diversity. And this is just because the sequencing machine was used, different sequencing machines, like different technologies, like different source of cells, etc., etc. Yeah, and T6 is not the time point of interest. If we go to the setup of the study, T6 is just somewhere, 1st of October 2021. 1st of October 2021, while the second COVID is like 17th of January 2022, or COVID-1 is the 15th of April of 2021. So it's completely unrelated. This is not our biology, but this actually will tell us very, very good thing about the downstream analysis we're going to do. So before we go to the downstream, one more just general overview how you can look on the repertoire. You can do other repertoire characterization like gene usage analysis to see what we have on the gene usage, on the V genes or J genes. You can also configure these visualizations into different ways and group by the metadata time points, change the colors, etc., etc. And we have a lot of other methods to look and characterize our repertoires. So now let's go and find which are those specific T cells that are activated with the immune system against the COVID. So looking at our study, we have T2 before the first infection, T3 after the first infection, and T8 after the second infection. So we can use differential analysis to find what are those T cells activated here and what are those T cells activated here. We can do this using the differential abundance block available in the platform, which runs the differential kind expression, but differential abundance analysis to find those specific T cells. So we configure this block to use time point from our metadata. As a numerator of our formula, we're using T3 and T8, the time points after the vaccination, after the COVID event. As a denominator, the baseline T2, which is our data before the COVID. So once we run it, we'll have the very, very same, I think, familiar to many of you, volcano plot. But this volcano plot is not for genes, as we used to in the bulk gene expression, but for the clonotypes. So here we have our clones, and specifically right now we're looking T8 versus T2. Again, returning to our study design, T8 is after second COVID, and T2 is before first COVID. And here we have clones, T cells, that are differentially abundant in replicates, in this T8 versus T2. So those are our candidates that we expect are the T cells activated with the COVID. So we can also compare them and see them on the, for example, multiple sequence alignments to see whether there are some patterns or motifs, and do things like that. So once we and we can check the same for T3, but for T3, it's actually not that visible as for T8. So T8 is more like, and actually the patient had a more severe condition at the second COVID compared to the first one. So yeah, with this information, we can, for example, now go to some nice visualizations. And for visualizations, we have a super powerful block. It's called GraphMaker. Yeah, GraphMaker, let me add it and show how you can configure and get to really, really nice plots. So GraphMaker is a very powerful tool where you can integrate all sources of the data and put on a very nice visuals. For example, for time tracking analysis, we are going to use stacked bar plus stream area plot. Let's select it. What we want to put on the y-axis. On the y-axis, we want to put the abundance of our clone. So the abundance is measured in the number of UMIs, so fraction of UMIs is the abundance metric. On the x-axis, we want to use time point. That's what we're going to use on the x-axis as a primary grouping. Yeah, and finally, we also want to look only on the clones that are differentially abundant here. We want to filter the data to the clones that are differentially abundant here. And let's do this by using the data generated, so this differential abundance block generated as this clonal type T8 versus T2 column that we can use here, select upregulated, meaning expressed or abundant at T8, yeah, as a filter. Nice. And the last thing here is to use CDR3 amino acid sequence as a secondary grouping. Boom, boom, boom. Yeah, let's also change the title to, for example, COVID associated TCRs, yeah, just for example. Let's also change the color to the, I like this one. Yeah, one more thing, since we're dealing with fractions, yeah, we also need, and this is a bar plot. Each bar should not be a sum, but a mean of the abundances of the individual contributions. Yeah, and that's it. And we have a very, very nice visual and nice and powerful biology here, because what we see the clones, yeah, that are expanded at the T8, yeah, and you see the resolution of the method. It's really great because we see they are differentially abundant here, and we don't see this on the clonality plot, for example, yeah. And what we also see here, and that's really interesting, that they are also activated at T3. So when we're looking for the clones associated with the COVID, doing differential analysis of the samples obtained after the second COVID infection versus the control, we also found the clones. We also found that they were actually also activated, but in the first COVID. And this is a very, very powerful and like quick way that take us 20 minutes, and then when you do it first time, even with the full data analysis, it will take like 1-2 hours to find the list of COVID-associated TCRs. Yeah, it's not a full list, and the paper, the authors went deeper and discovered more of them, but that's already a big thing. This is already a big part of that study. So with this, I'm going to finish my live presentation, a short story, how you can get from the raw FASTQ files prepared with the Miltenyi TCR kit to the final result of a time tracking of 5 TCRs associated with the COVID and bring them to the plot, which you can export and include in your publication, for example. Yeah. Returning to the slides, Platforma is available completely free for academic research, and yeah, here is some information how you can access Platforma and how you can access Miltenyi kits. Anything else to add, Monica, Yana?
Yes, absolutely. So we'll send the information out tomorrow, but just in case, you want to get a head start on the kits or Platforma, here's information here. So now we'll move into Q&A. So I saw one question that Yana had answered, but I just want to read it out for everyone. So this question comes from Martina. Does the presenter library prep and target enrichment workflow have the IVDR certification?
Yeah.
So for both the products and also for the analysis portion, it's RUO, so research use only. But if you are interested, you can reach out to Yana to discuss integrating the product into their IVD workflow. So if you have any other questions, feel free to drop them in the chats. So we'll be here for the next couple of minutes. And then we also had a question from Mustafa about which companies were in the competitors that Yana had showed. So she cannot disclose them, but if you have any questions, she has left the technical support email. So you can also reach out to her and if you have any issues with your clonotyping.
Mm-hmm. Yeah, I see other questions coming. So one of the question is how can we plot number of insertions, deletions in TCRs? So yeah, this information is available here. It depends on the configuration of the clone typing block, but basically let me try to do something. Let's go to the box plot and let's here, for example, number of reads, number of units. Yeah, number of edit nucleotides. It's here, total number of edit nucleotides. So we use it on the y-axis and let's then use again, for example, I don't know, best V gene as a grouping and see how many, I don't know, how many edit nucleotides we have associated with different regions, just for example, so you can play around with this visualization and with the capabilities of the visualization in many different ways to combine this. All the data is here. You just use it and play around with the types of visuals and what you can bring on the aesthetics and groupings and how you can layer this information to the space. Let me show you how you can do this on the clone type space, for example. Yeah, let's go to the clone type space. Yeah, and here you can, let's say, total number of edit nucleotides. Yeah, the edit nucleotides is exactly the ends. And let's put it as a color, for example. Yeah. So now our entire space of TCRs is colored by the number of edit nucleotides in the CDR region. Yeah, so just that's the way how you can visualize this data and plot it.
Great, thank you. So now we have a few questions from Yana. So I'll read the first one from Justina. Can this kit be used with total RNA isolated from hippocampal tissue that contains infiltrating T cells rather than from purified T cells or PBMCs? And what is the minimum proportion of T cells required in such a tissue sample to reliably detect TCR alpha beta transcripts?
Right. So the beauty of this question is that we have so different samples with so different varieties of immune cells and immune cell infiltration in this case, right? Our kit was tested with as low as 100 cells of input, but that was sorted T cells. So in your case, it is the whole RNA. I would say that it was always beneficial to first detect the numbers of cells that you would think are in this tissue, to then have a rough estimate, to then produce bulk RNA sequencing, but we can also do quality control steps in between. So if we're in close communications here, I, as Monica already said, put the technical support link here where we can link up and discuss the details of that. I think there's nothing hindering you to try out and see because it's so sensitive. An additional question is, I think I saw, do you happen to also have the BCR profiling kit? Not yet, but stay tuned. I hope that we can give you a positive answer to that soon.
Great. And then we also had a question from Mar in the chat. Do you have a minimum DV 200 of FFPE samples to be able to do TCR clone typing? And what kind of sequencing are you doing, short read or long read?
Should I answer?
Pardon me. Yeah, I thought
Yeah.
you were reading the first one in Platforma. No, it's short read, and we do it's 150 base pair, right? We're focusing on the CDR3 region here, and it is compatible with Illumina for now.
Great. And then I think this is our last question right now. So from Artem, he asks, is the prediction of TCR specificity also available in Platforma?
Sure, let me show you. I know, but by the way, when I was answering about the edit nucleotides, I was sharing my screen, yeah?
Yes, you are.
Okay, good, good. Okay, so for TCR specificity, yeah, we actually have a detect block from our partners. It's a company called ImmuneWatch. That's very nice algorithm for predicting TCR specificity based on the databases and machine learning models. It's available on the platform. You can add it and to use to predict TCR specificity and also HLA, yeah, from the data. So that's the answer.
Great. Keep your screen shared. As someone new to RNA-seq analysis, can we upload pre-processed data into Platforma for further downstream analysis and visualization of the data?
Yeah, sure. For bulk RNA-seq, I think we have, maybe I have it here. Yeah, bulk RNA-seq. Bulk RNA-seq, we have support of bulk RNA-seq analysis. You can both start with the raw FASTQ files, or you can upload the count matrices. Both options available. And then for the bulk RNA-seq, we have the standard pipeline of differential gene expression analysis where you can configure. It's based on the DC as well as all of the blocks in Platforma. They are based on well-known tools. Yeah, so you can do differential gene expression analysis, pathway enrichment, functional analysis to find the pathways and associated with the upregulated, downregulated gene list, etc.
Great. And then someone had also asked, how can we plot insertions for each samples for example? So she says she has tried this, but samples don't appear in a list to add an X.
Yeah. That's a tricky question for this. The raw ways how to do this, please contact our support team and they will provide you the solution offline. I can't show it right now. I don't have this data, but there are certain limitations, like number of edit nucleotides is a property of clonotype. It's not a property of sample. So what you want to do, like technically, when you're visualizing this, you're filtering the list of all clonotypes from all samples to the clonotypes that have more than zero, higher than zero, non-zero abundance in a certain sample, and then you're visualizing their CDR like number of insertions. So this sample association with the clonotype is slightly tricky. That's why we can provide you the way offline. So if you can share support email. Yeah.
Yep, I will. Agnes had asked a question about single cell, so I will just answer your question. But of course, today's focus is not about 10x. So yes, you can process single cell RNA-seq datasets with 10x or any other chemistry with Platforma. But if you want to learn more, just feel free to reach out to our support email. And Yana, so big question for you. Why should I be using Miltenyi's TCR profiling kit? What are the major advantages?
I love that question because I hoped to already have carved out the key advantages, but let me repeat again that we have a wide input range, like from zero 0.1 nanograms of RNA starting, flexible workflow, which allows to start from cells, skipping the step of RNA isolation. We further are able to work with FFPE samples compatibly, also exceeding other competitors, allowing cutting-edge science results also with complex samples. I mean, I could go on and nerd out about all the different things, but if you have a specific research question, I can go more into detail about why that might be helpful, maybe later on as well.
Great. And a question from Mar, just a follow-up. How many samples can be run at the same time, and how deep do you sequence? What is the number of base pair reads you aim to?
So let's go one by one. What was the first one?
How many samples can you run at the same time?
Right. I mean, technically, you can do the kit has 24 samples, but I would do 8 per because you always have to split into alpha, beta, and the handling would just get a little bit more complex. Yeah, that would be the max for handling it by hand as a single